Allora, ringrazio Paolo. Per me è un piacere così. Lo scopo del mio seminario è dare un'idea dell'importanza degli interferoni nel contrastare le infezioni virali, e quindi ho scelto questa tematica perché mi sembra molto attuale e sicuramente molti di noi si stanno convertendo sullo studio di questo virus, perché probabilmente ci darà molte informazioni di base, come sempre ha fatto lo studio sui virus, sulla biologia di base delle cellule. No? Ricordo per esempio che attraverso la virologia sono stati scoperti gli enzimi di restrizione, la trascrittasi inversa, gli oncogeni, il tumor suppressor factor, and so on. Passo all'inglese, così comincio il mio seminario. And the title is Viral Immune Evasion, the Interferon System, Coronavirus 2. And the first slide that I'm unable condividere, interrompi, sai, nuova condivisione. Vediamo se mi fa vedere, perché... ecco, vediamo un po'. Okay, I hope, okay. In the first slide, I can remind you that interferon are produced very early during acute viral infection, and this is a very old slide from people Baron and Bianzani. And as you see, interferon production with other innate immune defenses start very early. Very early, that means few hours. And it's really important to contrast the multiplication of the viruses and to give time to the host to develop innate adaptive immunity. So interferon were discovered in 1957 by two virologists, Isaacs and Lindemann, that were studying interference. And what they observed was that if you try to treat chorioallantoic membrane with inactivated influenza virus, the cell sends the virus and produce something in the medium that is able to confer resistance to viral replication after a few hours of incubation to a living virus, for example, a wild-type influenza virus. So cells of all the vertebrates feel the presence of the viruses and produce antiviral factors that are released outside to protect bystander cells. So what about interferon today?
Elisabetta, sorry to interrupt you, but we don't have a full view of your slides. Noi le vediamo per intero. Se metti a schermo intero.
E ricondivido. Partecipanti, condividere schermo. Is it right? Va bene?
Okay, perfetto.
Okay. So interferons are antimicrobial, antituberal, immunoregulatory cytokine, and they are subdivided in three antigenically unrelated groups. That is type 1, and I refer here to the human interferons because this molecule are specific. In the humans, there are 13 interferon alpha subtypes that are 65% correlated in amino acid sequences. Interferon beta, that is only one, it is 35% correlated in amino acid sequence with the interferon alpha type, but also other type of interferons. And all the gene is in the same chromosome because they derive during the time by viral duplication. Type 2 interferon gamma is only one, but is produced only by a restricted number of immune cells, mainly Th1 cells and NK cells, and interferon gamma production is a marker of the development of Th1. And in addition, NK cells produce interferon through sensing for example, the production of type 1 interferon. So the stimulus that induces interferon gamma production is antigenic or mitogenic. And this type of interferon is produced both later in the adaptive immune response and earlier during the innate immune response activation. Type 3 interferon is interferon lambda, was discovered in 2003. There are 4 subtypes in human, and the gene are on the same chromosome, but the receptor for this type of interferon is restricted to only few tissues, in particular epithelial cells, liver cells, and some immune cells. And the main role is counteracting pathogen at the front door. They are emerging as a key player in mucosal immunity. So I would like only to add that type 1 interferon is instead produced by all the nucleated cells when they sense pumps. Oddio, non mi va avanti. So, c'è qualcosa che non funziona. Dunque. There is something that doesn't work. Oddio, devo interrompere un'altra volta. Scusate, ma non so perché succede qualcosa di strano. Allora, proviamo a uscire un attimo, riproviamo qua. Allora, vediamo un attimino se mi riesce. Allora, ripartiamo dall'inizio. Condividere lo schermo, vediamo un po', condividi, allora, I don't know why there is something that doesn't work, so I have to...
Noi vediamo la presentazione.
Sì, però mi si è... Ah, ok. Benissimo, vediamo un po' se funziona. So, how they work? They work, all the 3 interferon types work in regulating hundreds of genes that are transcriptionally regulated and are named ISG, interferon stimulated genes. And in 2013, Paul Herzog set up an update database of annotated interferon-regulated genes, version 2, to help researchers that work in the field of interferon, because, as you can see, it's a very complex system. So the main signal transduction pathway that use interferon are the JAK-STAT. And this transduction pathway consists of receptors that bind a ligand and are recruited, and then Janus receptor tyrosine kinase are activated by recruitment, and then this Janus kinase phosphorylate the receptor chain, and the receptor chain recruit STAT, they are signal transducer and activator of transcription, so transcription factor. So it's a very simple signal transduction pathway, very direct, very rapid. And when STATs are phosphorylated, they homodimerize or dimerize or heterodimerize and become able to go inside the nucleus. So the signal transduction pathway is used by a lot of cytokines, but the JAK and the STAT are only few. So it's inside the cell that there is a combination of response that depends on what the cell sends outside. Regarding the interferon types, there are 3 receptors, one for each interferon type. They are different receptors, but use common signal transduction element. In particular, the STATs that are activated by this signal transduction are STAT1 and 2 for type 1 and 3 interferon, and only STAT1 for interferon gamma. And this is the main transduction element, because depending on the cell type or the tissue, it's possible that other starts come activated. In this way, you can see that type 1 interferon, type 3 interferon activate two main transcription factors. This one that is STAT1, STAT2, Irf1, and this one that is a homodimer STAT1. Instead, interferon gamma is able to activate this one. And here are named the enhancer element that are activated by the binding of the transcription factor to the interferon stimulated genes. The other thing that I would like to add is that STAT2 is the only STAT that is activated by type 1 and type 3 interferon, and there is no other cytokine that is able to induce STAT2 phosphorylation. So STAT2 is specific for the interferon signaling pathway, and in particular for type 1 and type 3. So this is a simplified schematic signaling cascade that refer only to type 3, type 1, and you can see that the transcription factors that are activated very similar. So the response to these two types of interferon is quite similar, but is also modulated by the additional element and other secondary signaling cascade. Another important aspect that we have to remind is the characteristics of type 1 and type 3 receptor. Type 1 receptor, these are the two chains that are recruited by type 1 interferon, are expressed in all the nucleated cells. Instead, for type 3 interferon, we have two elements. One is specific for interferon lambda, that is interferon lambda receptor 1. And the second chain is one chain that is used also by the interleukin 10 family. Interleukin 10 is an anti-inflammatory cytokine. So if the cell expresses this type of interferon of receptor, it can respond to interferon lambda because interferon lambda binds to these two molecules. And interferon lambda is also able to induce the production of interleukin 10. And when interleukin 10 is produced, recruit this receptor together with the other one, that is interleukin 10 receptor 1, subtracting this receptor to interferon lambda, thus down-regulating the signaling. Okay. Here, just to let you know that there is the canonical signaling that is that one that I have now described, but there are also non-canonical signaling that are more tissue-specific, cell type-specific, and so on. And I have no time to go inside this aspect. So I have told you that interferon-stimulated genes are a lot, hundreds. And to simplify the situation, I would like to divide the interferon-stimulated gene into main categories. The effectors, so that one that transcriptionally induced by interferon that bind to the receptor, act as antiviral molecule. And the other that are regulator. That means that one that regulate the immune response and also the appearance of other antiviral functions. Here I mention only some just to have an idea. And for example, regulator are the ability of all the three interferon types to increase the expression of class 1 and class 2 major histocompatibility antigen, thus facilitating the recognition by the adaptive immune response of the antigen. The increase of Fc receptor that increase the phagocytic ability of phagocyte, but also activate the ability of NK cell to respond to kill in a ADCC-dependent way cell, recognizing them in a specific way. And then I mentioned the Irf transcription factor that are less known, that are interferon responsive factor, a transcription factor induced specifically by different type of interferon. They are 9, and they regulate also the transcription of additional genes. For example, Irf1, that is a very famous one, induce the transcription of the inducible nitric oxide synthetase. Then I would like to describe some effectors. I only limited my description to 6. The first 2 are very well known because they were identified at the time when interferon was discovered, and are 2 double-strand RNA-dependent enzymes. So they, we can think that they are the additional double-strand RNA sensor that the cell do only if interferon induce their expression. And these 2 enzymes are able to induce inhibition protein synthesis, PKR, or degradation of messenger RNA if they detect double-strand RNA during viral replication. So if you treat cells with interferon, and then you infect with, for example, an RNA virus that is particularly sensitive, for example, the Ciclo virus, the cells survive because these 2 enzymes induce the inhibition of viral protein synthesis and degrade RNA in a very specific manner only where the virus is replicating, and the system is done regulating if the replicating RNA disappears. And so these 2 enzymes are very important to control viral expression. And so there are a lot of viral inhibitors of the PKR pathway. For example, I show here a slide just to let you know how many viruses can inhibit with different protein and different approach, and also via the production of double-strand RNA analogs, the activation of the PKR system, and the same is true also for the 2,5-A synthetase system. So, few words on these other 4 proteins that are particularly important in restricting the retrovirus replication, and HIV, that is very successful virus, is able to counteract their action. What they do, TRIM5A induce untimely uncoating of the nucleocapsid of the retroviruses, and so reduce the infectivity of the viral particles that enter in the cell. Apobacter G is a cytosine deaminase, and it's particularly important to reduce the infectivity of retroviruses because it acts during reverse transcription, inducing cytosine deaminase conversion of cytosine in uracil, and this induces as a final effect a G2A hypermutation, in reducing the infectivity of the proviral genome that is producing presence of these proteins. And in addition, it can also, the result of this, the action of this enzyme is also able to reduce the efficiency of the retrotranscription. So another one is tethering. Tethering is a membrane protein, and interferon-induced tethering. And for example, in the case of retrovirus, the tethering molecule is also acquired by the membrane of the budding virus. So one tethering molecule on the budding virus tether the other tethering molecule on the cell, and so the cell take up again the virus that before was budding and destroy the virus when the is internalized inside the cell. So in this case, HIV is able to counteract with a specific viral protein, VPU, the action of tethering that is highly increased by all the 3 types of interferon. So, and last but not least, ISG15, that is an ubiquitin-like modifier in use by type 1 and 3 that impairs the retrovirus budding process, inhibiting the interaction between the viral protein gag that guide the budding of the retrovirus, of all retroviruses, and the cellular protein that is involved in the vacuolation process in the cells. So, you have to understand that restriction factor, viral restriction factor induced by interferon to counteract viral infections are a lot and very effective. So many viruses evolve strategy to avoid the interferon system. But how interferon production is induced? This is a schematic model that describes how type 1 interferon genes are induced when the virus go inside. RNA and DNA, but also other microbial molecule are sensed by sensor, and this sensor activates the signal transduction pathway that activates IRF3, that is the main transcription factor that is responsible for the transcription and induction of interferon beta, activate also NFKB, and so we have the production of interferon beta. Then interferon beta is released, bind to the bystander cells, and regulate gene expression very early in a couple of hours. And I told you that many genes are upregulated, and between them there is another IRF molecule, Irf transcription factor, that is IRF7. And so now if a virus comes and the cell sense, activate both IRF3 and FKB, but also IRF7. And this IRF7 heterodimerize with IRF3, and this signal is able to induce the expression of the type 1 interferon gene. So there is the activation is a cascade of events. First, interferon beta is produced, and then interferon alpha produced. Interferon beta production is reinforced. The main difference between these two promoter activation is that this one is sensitive to the heterodimer. Interferon beta is sensitive to the homodimer and also to the heterodimer of RF3, RF7. And all interferon beta are regulated also by NF-kappa B, whereas instead interferon alpha is not. But in addition to interferon, the activation of I-kappa B can also have as a consequence the production of inflammatory cytokines. So how the cell sense the virus? And I restrict here my description only on two sensors because there are a lot of sensors, sensor of double-strand RNA, single-strand RNA, double-strand RNA, and so on, and you probably know that. And I would like to restrict to double-strand RNA sensor. We have here, I show here toll-like receptor 3 that is present in intracellular vesicle inside the cell, and also the family of the double-strand RNA sensor that are named Regai-like. And what they work. Regai-like sense the presence of double-strand RNA made during viral RNA replication. Obviously, is a particular double-strand RNA. It needs to have particular characteristics, and it is not required to be big because it's enough 20 nucleotides of double-strand RNA to activate this type of enzyme. And the double-strand RNA is bind to the helicase moiety of this sensor. So the sensor change the conformation and expose these two elements, the card element, and this card element tether the card element of the mitochondria antiviral protein MAVS. And what happened at this time is that MAVS form a prion-like structure that is required for the activation of this type of a kinase that induce the activation of IRF3. And also NF-kappa-B activation is obtained, so interferon beta can be produced. The other pathway is different from this one because do not require the activation of MAVS protein, but involve other adapters. But at the end, the activation is similar. So activation of NF-kappa-B and of IRF3 family. So what about Coronavirude? Coronavirude, as you well know now, is a single-strand RNA positive virus. They are lytic viruses, and the name of the order is Nidovirales. And this order is composed of some family of RNA virus that are characterized by the ability to form nested RNA transcription during the replication. And almost all alpha and beta coronavirus have mammalian hosts, including humans. And many coronavirus are important veterinary pathogens, for example, feline infectious peritonitis virus and avian infectious bronchitis virus. And the last virus is a very important veterinary pathogen because it reduces the performance of good meat type and egg-laying chickens and cause severe economic loss to the poultry industry worldwide because it is a highly pathogenic virus. And in the case of human, we have 7 human coronavirus, and 4 are well known and identified before the SARS-CoV-1 appearance, and they are responsible of only upper respiratory tract infection and account for a significant percentage of adult common cold. And the other 3 are severe acute pathogen that induce severe acute respiratory syndrome. And the particle is this is an electron micrograph that show the crown-like corona that characterize this picture. And the coronavirus particle is characterized by club-like projection on the surface, which are heavily glycosylated and formed by trimeric spike S protein, an abundant membrane protein that give the variant shape and is also able to guide the budding process because it's also able to induce the curvature of the cell membranes. And in the membrane, there is also an E protein that is a channel that an essential role during assembly. And also sometimes there is also another enzyme that is not present in the SARS-CoV-2 virus. And inside we have an helical nucleocapsid that is formed by the binding of the nucleocapsid protein that bind the genomic RNA in a bits on a string fashion. So the genome size of coronavirus is quite big because it's the largest in the RNA virus world. So these are quite complex virus in the RNA virus world. I would like to remind you that HIV is 1/3 the genome of HIV is 1/3, the genome of hepatitis virus is 1/3 the one of the coronavirus family virus. And what about the life cycle? There is an attachment entry, infection start with receptor binding, and the major receptor for SARS-CoV are, as you know, ACE2. And the S protein is cleaved by host protease into functional subunit, the N-terminal receptor binding S1, and the C-terminal subunit responsible for membrane fusion. And this is the genomic organization. Here I show you a picture from the prototype virus of the family. That is the mouse hepatitis virus, but just to have an idea of the organization of all the viruses of this family. So we have this RNA molecule that is capped at 5' and a polyA sequence is present at the end. So it's a typical eukaryotic messenger RNA. And so translation starts immediately, and a first precursor is produced, the protein 1A, and then translation stop and restart from the 5' ends. And sometimes there is a frame shift here, and via ribosomal frame shifting is produced another precursor that is bigger, this one. And then again, translation stops and is unable to go further. So from these two precursors, 16 different proteins are produced, and obviously there is the RNA-dependent polymerase, some enzyme that provide some proofreading activity of the enzyme, and also other ones that are able to form complex with this enzyme to regulate transcription. Obviously, in the precursor protein are present also two proteases that are responsible for cleaving the precursor. This one is a papain-like protease, the NSP3 protein. And the other one is named the main protease because it cuts more site than the other one, the precursor protein. What happened thereafter? Replication and transcription coronavirus involved the formation of replication transcription complex anchored to intracellular membrane via multi-spanning transmembrane protein, viral protein, the SP3, 4, and 6, for example. And inside the infected cell, coronavirus induced important modification of the intracellular membrane network, forming double-stranded double membrane vesicle. And several studies have shown that double membrane vesicles are closely associated with the coronavirus replication complex. And using the genomic RNA as a template, replicate synthesize the negative sense genomic, but also use this template to produce the progeny positive sense, but also through discontinued transcription, produce a set of nested sets of genomic RNA, because the RNA polymerase is able to jump from during transcription through this particular conserved sequence that are named TRS. So this is a schematic presentation of the SARS-CoV-2 genome. This is the genomic RNA, and these are the subgenomic positive RNA that are all coupled and have a polyA sequence, and these modifications are realized in the cytoplasm because the replication of this virus occur in the cytoplasmic compartment. And as you see, this subgenomic RNA serve to produce the structural protein M, A, N, but also other accessory proteins that are named ORF6, ORF7, and so on, that regulate the late phase of the cycle. And here I show you an electron micrograph that show that coronavirus are able to induce membrane alteration inside the cell and the formation of these multiple double membrane vesicles that are characteristics of the replication of the virus. And so in the late phase, after production of the structural protein that are synthesized and inserted and folded in endoplasmic reticulum and transported to the ergic compartment, there is also, it begins the viral assembly that occurs at the ergic, so at this level, it is likely to be orchestrated by the M protein, the protein that is the main protein present in the envelope and that shape the virus. So the virus budded into ergic are exported through secretory pathway in smooth wall vesicle, which ultimately fuse with the plasma membrane and release the mature viral particle. So here with this slide, only to show you example of an RNA virus that antagonize the interferon system, because obviously the interferon system contrasts viral replication at a lot of levels. And what about coronavirus? All the coronavirus contrast the interferon system, but in particular, is particularly able to contrast the interferon system, the highly pathogenic coronaviruses that use various approach and different viral protein to counteract the system, inhibiting production, signaling, and so on. For example, in the case of SARS-CoV-1, this protein is involved in contrasting the interferon signaling. And there is a picture that shows the 16 non-structural protein coronavirus, and what we know, we knew before SARS-CoV-2 entered in human population. So there is a lot of proteins that are involved in contrasting the interferon signaling. And these proteins are also involved, have multiple functions. So they have required both to carry out replication of the virus and are involved in evasion of the innate immune response. So what about SARS-CoV-2? Now I would like to show you some data that has been published in the literature, because there are many thousands of papers on SARS-CoV-2. And this is one paper that is particularly interesting and was published in October in Cell Reports, in which the author performed the screening of all the 27 SARS-CoV-2 proteins except one to identify those that antagonize the type 1 interferon response. And I show you what they have done. They have expressed through expression, single expression vector of single protein inside 293 cells each protein. And the only one that they failed was NS3. And then what they have done, they have transfected the cell with the vector that express each protein, and then they have co-transfected also a luciferase reporter vector that in which the luciferase is under the control of the interferon beta promoter, and they have induced also they have used another vector that give an activation signal starting from RIG-I, and have looked which of this protein is able to inhibit the activation of interferon beta promoter that I've shown you is the first that is activated sensing virus. And so they have identified 4 proteins that are able to do that. 4 proteins that are NSP1, NSP6, NSP13, and ORF6. ORF6 is a late protein. And using an approach that I have no time to describe, they have also identified what is the step that this protein contrasts. And with a similar approach in which they contrast factor the single construct that express each protein with a reporter luciferase construct that express the luciferase under the control of the enhancer present in the interferon-induced genes. They also identified which proteins inhibit the type 1 interferon signaling. And as you can see, we can summarize this result in this way. We have an NSP1 that suppressed type 1 interferon system at multiple levels because it induced cellular messenger RNA degradation and translation inhibition of host genes. Then we have an NSP6 that binds TANK TBK1 kinase and suppress RF3 phosphorylation, but also blocks STAT1 and STAT2 phosphorylation. And SPIN13 binds and blocks TBK1 phosphorylation, and blocks also STAT1 and STAT2 phosphorylation. ORF3A blocks STAT1 phosphorylation. ORF6 bind to a particular important carioferrin alpha-2 that is responsible for nuclear translocation of both IRF3 and also STAT1. So ORF6 block the induction of interferon production, but also the interferon signaling. And ORF7 blocks STAT2 phosphorylation, ORF7B blocks STAT1 and STAT2, and M blocks STAT1 phosphorylation. So a very complex inhibition of the interferon pathway that is inhibition not only of the type 1 interferon signaling pathway, but probably is also an inhibition also the interferon lambda signaling pathway because STAT1 and STAT2 I have shown you before that are involved. So there are also other papers that is coming out that confirm this result with some different, why the virus needs to block not only the production of interferon, but also the mechanism of action. Because in vivo, when the virus infects the cell, a swarm of viral particles are produced, and this swarm of viral particles is full also of defective viruses. And are the defective viruses that, for example, have some defect in the ability to block interferon production that in vivo induce the production of interferon. So interferon is produced after viral infection, in any case, also if the virus has the ability, the RNA virus have the ability to induce, to block their production inside the cell, because to block it need to be a wild-type virus or a virus that have no mutation on the proteins that contrast the interferon pathway. So, and what the author did also, they set up a SARS-CoV-2 luciferase replicon that allow them to study the effect of interference of viral replication in vitro and the ability of the viral protein to counteract this specific action. And this replicon is particularly useful because, as I summarize here in the highlight of the paper, SARS-CoV-2 protein antagonizing interferon production and signaling with different proteins, but the luciferase reporter replicon SARS-CoV-2 will allow experiment at biosafety level 2 to study the replication process of these viruses is quite complex, and will be also useful to test antiviral molecules. So, how much interferon is important to avoid the development of highly pathogenic phenotype? From the past, we have observed that when the virus induces a pathogenic dysregulated inflammation, there is a robust virus replication, instead of doing robust virus replication, but there is also a delayed interferon response. Instead, when the response to the infection is protective, you have mild symptoms, you have early interferon response. And the delayed interferon response is accompanied by an inflammatory monocyte macrophage and neutrophil infiltration and pro-inflammatory cytokine chemokine production, so the famous cytokine storm. But one can ask if the delayed interferon response is really important for the pathogenic phenotype. And this is the case because two studies recently examining the likelihood that genetic affects the risk of severe coronavirus disease through component of the interferon system. And these are the two papers that came out last October in Science. In the first paper, it has been observed that people that develop life-threatening infection, life-threatening pathogenic effects, in this cohort of 1,000 people, 10% have neutralizing autoantibodies that impair type 1 interferon immunities, that are antibodies that neutralize interferon alpha and interferon omega. Instead, in the people that develop asymptomatic or mild infection, and the sample was about 700 people, 0% have antibody that neutralize type 1 interferon. So this is a strong indication that the blockability of this antibody does not help people that have that problem before the infection, increase the susceptibility of these people to develop a life-threatening pneumonia. And the second paper that came out in the same journal and in the same paper is that inborn errors of toll-like receptor T and Irf dependent upon interferon production and amplification underlie life-threatening COVID-19 pneumonia. In fact, in this study, they found that at least 3.5% of patients with life-threatening COVID-19 pneumonia had known deficiency that involved the toll-like receptor 3 and Irf dependent induction and amplification of type 1 interferon production. So for this reason, some people retain that perhaps interferon beta could be useful for treat patients that develop COVID not with a mild disease, obviously, but you have to choose the right timing very early, using the right subtype, and the community think that interferon beta is the case, to ameliorate the pathology and have a mild COVID-19 and a viral clearance. And there are some clinical trials that are looking that are that are coming to try if this is true. But what is more important is what comes from the past, because there is a possibility that we have another approach that can restore interferon production. And this possibility comes from the result that we have obtained by basic science, studying the replication cycle of hepatitis C virus. This is an envelope RNA plus virus. The genome is 1/3 that of coronavirus, with only one Irf. So the RNA, when go inside the cell, produces a single polyprotein that contains two, that is cleaved by cellular but also viral protease. And so this protein are involved in the replication and budding and so on of the virus. And the hepatitis C, in the 75% of the case, induce a chronic infection that is often asymptomatic. And after some years, people develop liver damage with cirrhosis, and sometimes liver carcinoma develop. And what we have to combat this infection. In the past, until 2011, people with chronic infection that show some liver damage were treated with a combination of type 1 interferon, a ribavirin, that is an inhibitor of RNA polymerase. But with contrasting results, sometimes works, sometimes no, doesn't work, because it depends on the genotype, and different genotypes have different ability to contrast interferon pathway, so the result was not so good. But when an inhibitor of the NS3, NS4A protease was developed, all became better. And this inhibitor started the DAA revolution era. DAA is direct antiviral, direct-acting antivirals. And what does the inhibitor? The inhibitor blocks the maturation of this precursor, so it acts on the replication, but also this type of protease have another target, and the other target of the human hepatitis virus C protease NS4 is two proteins that are involved in the sensing of the RNA virus inside the infected cell. So when you use this inhibitor, you have two effects. The first effect is inhibiting replication, inhibiting the maturation of the precursor, and then we have also probably the restoration of type 1 interferon production at the site of infection. So you avoid the problem of use interferon in therapy, because when you administer interferon, interferon go interact with all the cells of the body, so you can have side effect. Instead, this molecule act physiologically at the site where they are induced. And so I think that I can finish my seminar, hoping then trying to find inhibitor for SARS-CoV-2 viral protein that are involved in contrasting the interferon production. Possibly we could develop effective antivirals that could be used in case of NIDS, and is another strategy to combat, in addition to vaccination, is obviously is absolutely necessary. So I finish my seminar. Thanks for your attention, and I hope that I give you an idea of the system.